Our journey through the mutations still continues. We saw the various gene mutations, the what are the different types of gene mutations, what are the causes of gene mutations. That is all studied in the previous classes. Now today we're going to talk about the different chromosomal mutations. Mutations, as we told, are sudden, randomly occurring heritable changes within, let it be in microbes, in humans, or in different plants as well as in the animal forms. They can occur for various reasons, either for the evolution or it might be destructive to the species. Okay, so now other than be occurring in the gene level, sometimes what happens, the mutations when it occurs in the chromosomes, certain changes happens to the chromosomes. And these changes can be sometimes be in a numerical order or a structural variation. So based upon this, the chromosomal mutations are mainly divided into two types. That is the numerical mutations as well as the structural mutations. The numerical mutations, it mainly includes changes in the number of the chromosomes within a microbe or a eukaryote or any other form. This mainly occurs due to the irregularities in the nuclear division, and as a result, what happens? The cells of the organisms will have a changed chromosome number and they will be found to be heteroploid. Okay, that is, they have a different chromosome number. The number ploidy, it refers to the number of chromosomes. Okay, so unlike the wild organism or wild strain, if we are talking about the microbe, the chromosomal mutations which involve the numerical changes will result in the formation of heteroploid organisms. Now, the structural variations due to chromosomal mutations, these can occur in both plants and the animals, and they result in increased variation, reproductive isolation, and thus evolution to happen. And there are different types of structural variations such as deletions, duplications, inversions, and translocations. Now you might think, like we had already spoken about deletions, duplications, inversions, etc. in the case of genes. In the case of chromosomes, the same theory applies here, but the thing is that it would be in a very higher range, that is in the chromosome, rather than occurring in a gene. So when the mutation, structural variations happen in the chromosomal structure by various processes such as deletions, duplications, inversions, and translocations, the chromosome undergoes various structural changes. And those structural changes will result in the formation of various mutations as well as various phenotypic characters as well as the various genotypic characters of the organism gets altered. Now, when the number of, coming to the number of the chromosomes, when the number of the chromosomes changes, we say that, say that they become heteroploid. And there are mainly two types of heteroploid, that is chromosome number mediated variations. It can be euploidy and aneuploidy. In this session, we will be talking about euploidy. Let's go into the details of that. Euploidy or polyploidy is a situation in which you are getting an entire set of chromosomes. Euploid means it's a true or a even possessing entire set of chromosomes or the genomes. If you say, now when you consider to be polyploid, what happens? You say that you get one whole set of chromosome is being duplicated. For example, here, if consider 2n is equal to 6, that is a 2x. Okay, if it becomes a triploid, okay, that is referred to as a 3x, and 4x refers to a tetraploid. So it means that when a mutation will result in the formation of increase in the entire set of chromosome, or one chromosome itself, when it has been multiplied, when when the entire set of chromosomes are getting multiplied, that is referred to as a polyploidy. Now, if you have only one single set of chromosome, it is referred to as a monoploid. Now, If you have two sets of chromosome, you refer it as a diploid. And if you have three sets of chromosomes, it refers as a triploid. So you should understand that these two chromosomes are what? Are what? They are diploid. Okay, they have two chromosomes. And here also they have a diploid chromosome. Okay. Now, what is a polyploid? What do you mean by polyploid? Sometimes in certain cases what happens when, during the meiotic division, when it happens, what happens? The diploid parent species, which is having two, and these are three different chromosomes, and what happens? This is the parent species. And when a meiosis happens, what happens? The result is this homologous chromosome will result in the formation of what? It will split, okay? And the split, and it is supposed to come to an, it will result in the formation of what? Uh, N. That is 2N, it has to become into N. Okay, but in some cases, because of a due to some meiotic errors, what happens? Instead of becoming N, the gametes will remain as 2N. And as a result, self-fertilization can happen, and it will result in the formation of a zygote, which is being found to be 4N, or it becomes tetraploid. So what happens? The entire set of chromosomes is being multiplied. So a condition in which an entire set of chromosomes is being multiplied, that is referred to as polyploidy. Okay. So the homologous chromosome here, so we have to say the homologous chromosomes, they will fail to separate during meiosis, as the meiotic spindle will not be formed. Okay, you know the spindle fibers which are being formed during both mitosis and meiosis. They are the ones which separate the chromosomes into two directions. So what happens when the meiotic spindle will not happen? What happens? The diploid gametes will fuse with other diploid gametes here in this condition and result in the formation of a tetraploid offspring. And another condition where there would be also a failure of cytokinesis after the telophase stage of karyokinesis, that is after the nuclear division. What happens? The cytokinesis might not happen. So that also could result in the formation of increase in the whole set of chromosomes. This condition is referred to as polyploidy. And this is often seen in the case of plants, and in the case of animals, these have been found to be very much rare, but you can see it in some reptiles like salamanders, okay, and also in some goldfishes and all, you can see this polyploidy. So it is a condition where the entire set of chromosomes are being, the whole set of chromosomes are being increased. Okay. So we saw the reason why the polyploidy is occurring here. It was due to a meiotic error in which the 2N organism was not being converted into a 1N gamete. Now the normal condition is 2N or a diploid species will be converted into 1N gamete. Instead of that, here it remains as 2N itself. And so when they fuse, you get a tetraploid zygote. Now, polyploids, they can be divided into two types. One is the autopolyploids and the allopolyploids. Okay, when the same genome is duplicated by that is referred to as an autopolyploid. But if you consider when the chromosomes of two different species are being joined together, then it results in the formation of allopolyploids. Okay, now autopolyploids, you should remember the same species organism itself is having, the genome is getting duplicated. This can be by different conditions. Now it can be due to protoplast fusion technique, or it can be giving some drug like colchicine, which is a mitotic spindle inhibitor, or sometimes the egg itself is fertilized by more than one sperm. So as a result, what happens? The genome of both the sperms will enter into the zygote. So that would also be result in the formation of autopolyploids. Okay, so when a polyploidy occurs in the same species, that is referred to as an autopolyploid, and when it occurs between two different species, that is referred to as an allopolyploidy. Now, in autopolyploids, you can see that in some somatic cells of animals, such as we mentioned it earlier, in goldfish and salmon and salamanders, you can see autopolyploids. Now, what is an allopolyploid? Now, for example, we had mentioned earlier, uh, we need two different species to be undergoing what happens. That is, their genomes have to be joined together. Now, in the case of Raphinobrassica, okay, this was a yellow polyploid which was being developed, okay, and it is mainly done by the hybridization of two different varieties, okay. One is radish, okay, radish as that is raphanus, okay, and we have cabbage that is brassica, okay. These two join together and they both are having a diploid number of about 18. So 2n and 2n of cabbage and 2n of radish combines together to give another hybrid called the Raphinobrassica, which is 2n is which is also diploid, and but it has a this thing. It has a genome content of 2n is equal to 36. And the chromosome number is being found to be 36 here. And this species is a hybrid variety, which is like having the root of a cabbage and the leaf of a radish. So usually in the case of plants, okay, the polyploids are mainly being formed to either to improve its vigor, to increase the size and all that. But in the case of animals, it has been found to be not that much prevalent. And so another reason that we can have here is, now I had told you about the allopolyploids. That's what is being depicted here. So you have species 1 and you have species 2, and this is the normal gamete, okay, where it becomes the homologous chromosomes get split and you get single, half the chromosome, or the chromatid comes, okay. And then after that, after the mating, what happens here? The first mating is happening, you get a polyploid gamete, okay. This is a polyploid gamete which is having, and it will result in the second meeting, it will result in the formation of both the species together will combine together, and you will get a polyploid species. That is, species 1 and species 2 will be hybridized by different methods, and it will result in the formation of polyploid organisms. Okay. Now, this will go to the next version, polyploidy in the case of humans. True polyploidy is rarely occurs in humans, but you can see that in some cells, now for example, in the liver parenchyma, heart muscle, placenta, as well as in the bone marrow, we can find some polyploidy is being visible in the particular tissues. And sometimes polyploidy can occur in the form of triploidy. That is, instead of 2n, we might get a 3n condition, okay, with 69 chromosomes, okay, sometimes called the 69 or the triple X comes. And also tetraploidy also can happen in the that getting 92 chromosomes also is being found, okay. And that all these conditions, let it be triploidy or let it be tetraploidy, that will result in the formation of various genetical disorders. Now, in most of the cases, when you have triploidy, what happens, because due to polyspermy, I had mentioned earlier, one egg is being fertilized by different sperms. In such a conditions, for example, it will result to almost 2 to 3% of the human pregnancies will have a triploidy, and it will lead to 15% of the nature itself will remove it by a miscarriage. And the features of polyploidy, which we have, is they may become reproductively isolated from diploid populations if we are trying to make them. Okay, if we are artificially trying to make them, when the polyploid populations are being formed, they will be reproduced productively isolated. And this is more common in the case of plants than in the animals, because it is more likely to be fatal to the organism that have chromosomes for sex determination. And in some plants, as I told you earlier, sometimes it might result in a better phenotypic variation, and that is referred to as hybrid vigor. And in the case of Solanum tuberosum and Solanum brevidens, you can see that they get resistance to blight. So that's about polyploidy. We'll continue in the next sessions, the aneuploidy. Thank you.